The fundamental challenge in advanced medicine is safely delivering genetic material into target cells. Gene therapy using DNA holds immense therapeutic potential. However, naked DNA faces a critical biological barrier. Due to its large molecular size and highly hydrophilic negative charge, it cannot cross the hydrophobic cell membrane by itself. In addition, once DNA enters the body, it can be rapidly degraded by circulating nucleuses. To solve this problem, we need an efficient nano-delivery system. This system should mask the DNA charge, protect DNA from degradation, and help DNA enter the target cells. To achieve these functions, lipid nanoparticles, or LNPs, were used as the delivery system. LNPs are nanoscale carriers composed of lipids that can encapsulate genetic materials such as DNA. LNPs are formulates by mixing ethanol phase with an acidic aqueous phase, by forcing a self-assembly in a microfluidic system. They help protect the cargo during delivery and facilitate its transport into target cells. Because of these advantages, LNPs have become one of the most widely used platforms for nucleic acid delivery. To understand our experimental design, we need to connect three key points. These are total flow rate, the nanogenerator microfluidic mechanism, and DLS characterization. When lipid molecules dissolved in ethanol meet the aqueous DNA buffer, they undergo rapid nanoprecipitation. This happens because lipids are hydrophobic in water. The physical speed of phase collision is controlled by the total flow rate. This is why we use the nanogenerator FlexN. Conventional bulk mixing is slow and creates uneven concentration zones. This can lead to large and non-uniform aggregates. In contrast, the nanogenerator uses microchannels to apply precise kinetic energy and high shear stress. To validate the final LNP properties, we utilize a Zetasizer via dynamic light scattering. We do not use this instrument only to report numerical values. Instead, we use it to extract key biological indicators. Particle size and polydispersity index are critical measurement targets. If LNPs are too large, they may be cleared by the immune system. The Zetasizer measures Brownian motion and analyzes light scattering fluctuations. From this, it calculates the hydrodynamic diameter. We also measure zeta potential to evaluate surface charge. Surface charge affects colloidal stability and cellular interaction. By analyzing the charge distribution, we can evaluate whether DNA is mainly encapsulated inside or exposed outside the particles. This information helps us optimize the DNA delivery system. Phase 1 is sample preparation. Inside a fume hood, our 4-lipid matrix is prepared. The lipid components are DODAP, DSPC, cholesterol, and PEG-2000. These lipids are first dissolved in chloroform. To remove residual toxic solvent, chloroform is completely evaporated. This is done using a nitrogen stream and a vacuum desiccator. The resulting dry lipid film is reconstituted in 100% ethanol. Then, it is thermally incubated and sonicated. This produces a homogeneous organic phase. Separately, the aqueous phase is prepared. The target GPR plasmid DNA is mixed into filtered pH 4 buffer. The solution is kept in an ice bath to maintain molecular stability. After automated washing cycles, the lipid phase and DNA phase are loaded into the system. To map the fluid dynamics, we program 3 total flow rates. These are 2, 6, and 10 milliliters per minute. After LNP synthesis, the samples are loaded into a mini dialysis kit. This step removes free plasmid DNA and residual ethanol. The dialysis assembly is immersed in deionized water. It is maintained under continuous stirring at 4 degrees Celsius for 24 hours. The purified LNP samples are serially diluted in PBS. First, the sample is loaded into a standard cuvette to measure particle size and PDI. Then, the sample is loaded into a disposable folded capillary cell. This is used to measure zeta potential. This experiment is based on 2 hypotheses. These hypotheses are based on fluid dynamics and lipid chemistry. The first hypothesis focuses on DNA encapsulation. We expected that adding DNA would increase the particle size. At the same time, the final surface charge should remain close to neutral. This is because large DNA molecules are wrapped inside the ionizable lipid core. This naturally increases the hydrodynamic diameter. However, DNA is locked inside the particles and shielded by PEG lipids on the surface. Therefore, the final zeta potential should remain near neutral. The second hypothesis focuses on manufacturing. More specifically, it focuses on total flow rate. We expected that increasing the flow rate would first decrease particle size. This is because faster mixing creates rapid supersaturation. As a result, lipids precipitate quickly into smaller and more uniform particles. However, if the flow rate becomes too high, excessive kinetic energy and reduced residence time may disrupt proper self-assembly. This could lead to chaotic aggregation and larger particle formation. For zeta potential, we expected relatively stable values across different flow rates. This is because surface charge is mainly governed by lipid composition and particle surface chemistry. Therefore, flow rate should not strongly affect the final surface charge. Now, let's look at the comparative analysis data. We can check whether the formulation results match our theoretical predictions. First, we examined the effect of DNA encapsulation. The data supports the first hypothesis. Blank LNPs showed a smaller particle size than DNA-loaded LNPs. This suggests successful structural integration and encapsulation of DNA. The zeta potential showed only a slight shift. This small shift indicates that the negatively charged DNA was mainly located inside the particle core. It also suggests that DNA was effectively shielded by the outer lipid layer. On the other hand, the polydispersity index results show that the formulation with a total flow rate of 6 ml per minute achieved the lowest PDI value. This indicates the most uniform particle size distribution. Overall, all formulations showed PDI values below 0.3, except for the total flow rate of 10 ml per minute. The higher PDI value at 10 ml per minute was likely due to particle aggregation. Therefore, the formulations generally demonstrated good particle uniformity, with the total flow rate of 6 ml per minute showing the best performance. Next, we examined the effect of total flow rate on particle size. As the flow rate increased from 2 to 6 ml per minute, the average particle size decreased significantly. This matches our hypothesis. Faster mixing promotes rapid lipid supersaturation. This produces smaller and more uniform particles. However, when the flow rate increased from 6 to 10 ml per minute, the particle size increased dramatically. At 10 ml per minute, excessive fluid velocity may have disrupted stable mixing. Instead of controlled self-assembly, chaotic aggregation may have occurred. Finally, we analyzed the surface charge across different flow rates. The zeta potential remained highly consistent. This supports the idea that surface charge is mainly controlled by lipid composition and surface chemistry. At higher flow rates, enhanced mixing may produce more homogeneous nanoparticles. This may slightly increase the effective surface charge density. As a result, the zeta potential may become slightly more negative. In conclusion, the initial research goals were successfully achieved. The data supports successful DNA encapsulation inside the lipid core. It also shows that DNA was effectively shielded by the outer lipid layer. Furthermore, this experiment identified 6 ml per minute as the optimal flow rate. At this condition, small and uniform LNPs were produced. However, increasing the flow rate to 10 ml per minute exceeded the kinetic threshold. This caused chaotic aggregation and increased particle size. Overall, these results establish the operational boundaries for scalable and reproducible LNP manufacturing.